It's my pleasure to introduce my colleague, Mikhail Shapiro. But before I do that, I want to make a comment on each of the two presentations that are coming up. We are doing an experiment this time where we decided that you would not only like to hear from our faculty colleagues, but you might like to have a window into the laboratories where the work is done, and you might like to hear from the students or postdocs who are actually doing the work. And so there will be video presentations incorporated into the talks. And we have George Daglian and Hannah Manich here sitting in the back there. Maybe you could wave to us. Thank you. So you'll see them in the videos in just a minute, and they will be staying with us through lunch. So if you'd like to talk with them over lunch, you'll have an opportunity to do that. So now it's my pleasure to introduce Mikhail, who is my colleague in the Faculty of Chemical Engineering. And to give you some sense of the interdisciplinarity of the Caltech faculty, Mikhail has no degrees in chemical engineering, and neither do I.
We have other degrees.
Mikhail was trained initially in neuroscience at Brown University and then in bioengineering at MIT. He then spent 3 years at Third Rock in the venture capital world, and then 3 years as a Miller Fellow at the University of California, Berkeley. He joined us in 2014 and has developed an important research program in the area that he'll discuss with us using ultrasound and other methods to image within the body and also to intervene in a therapeutic sense. I would also point out that Mikhail is an investigator of the Howard Hughes Medical Institute. That is a very competitive kind of appointment and testament to Mikhail's research excellence. And in addition to that, I would like to point out that we have 8 HHMI investigators on the campus, and 5 of them were initially investigators of the Heritage Medical Research Institute. And you may recognize that as a result of the gift of our colleague, Richard Merkin. And so 5 of our 8 HHMI investigators had an early start as HMRI investigators, and shows the impact of that program in accelerating the development of research programs in biomedical science and engineering. That's been a wonderful addition to the Caltech campus. And so I'd now like to ask Mikhail to come up to the front and tell us about cells that speak.
All right. All right, thank you, Dave.
Thank you very much for having me here. It's a pleasure to be in front of this group, and thank you for everything that you do for Caltech. So let's get into it. We are a bunch of nerds near Hollywood, so it's no surprise that we take some inspiration from science fiction movies. And some of you may have seen The Fantastic Voyage, perhaps when it first came out. Sorry, that was low. All right. And as you may recall, it's a movie starring Raquel Welch, where the concept was that there was a nuclear physicist who had a blood clot, and to prevent nuclear war disaster, a bunch of scientists and engineers went into a submarine that then was shrunk down to the size of a cell and sent into the bloodstream so they could find the blood clot and resolve it, and happily ever after. And this is very inspiring. I think many of us at Caltech view this as like something that we'd want to create. But in our lab, we have a little bit of a different take on this, that instead of shrinking a submarine down to the size of a cell, we can just start with the cell itself and then engineer it to do submarine-like stuff inside the body. And the reason for this is because if you think about what that submarine needs to do, migrate through the bloodstream, find sites of disease, infiltrate into tissues, perform different kind of functions, cells already know how to do 90% of that. And what we want to do is take that ability and then equip these cells with a little bit of additional functionality that allows them to do our bidding inside of the body. And this is not something that we came up with ourselves. There's an entire field of synthetic biology And there have been some spectacular advances in the last decade or so. For example, with CAR T cells for cancer immunotherapy, these are immune cells that are taken out of the bloodstream of cancer patients, modified genetically so that they can recognize tumors inside the body and sent back in to fight and kill the tumors. And this is an FDA-approved therapy for some forms of cancer. You also have things like probiotics, similar to the ones we eat in our yogurt in the morning, that are now being engineered so they can go into the gastrointestinal tract and produce anti-inflammatory compounds, for example, to help with gut inflammation. And there's a lot of work and interest in stem cells, et cetera. And one of the views we have on this is that when we send these very sophisticated agents into the body, it would be nice to have a way to tell, where are they? Are they alive? Are they doing what we wanted? And ideally also tell them what to do based on their location inside the body. Now, doing that, talking to these cells today, is very challenging because the main way biologists talk to cells is using light. And you've probably seen these beautiful images of cells on the left and little worms in the middle and pieces of brain on the right, where the color in these images is coming from fluorescent proteins that cells can express that then allow them to show up under light. And the most famous of these is green fluorescent protein. This is used in biology labs throughout the world. And this protein came actually from jellyfish that naturally have this fluorescence in them. And the big breakthrough was when scientists took the genes from the jellyfish that allow them to make green fluorescent protein and started putting those genes into all these other kind of cells, like the human cells under the microscope, the mouse brain cells, et cetera. And this was recognized by the Nobel Prize in Chemistry. But there's a big problem. If we're trying to do this inside the body of even small animals and eventually humans, that's going to be very difficult because light doesn't penetrate well into the body. So these mice in this image, the ones on the left and right, have fluorescent cells all throughout their body, but you can only see the fluorescence like coming out of their eyes and through the ears because the skin and the hair is already enough to block all the light from coming out. So we're not going to be able to really do a good job of talking to cells inside the body using light. So what should we use instead? And we know that if you go into a hospital, instead of fluorescence looking at you, you'll have things like ultrasound and magnetic resonance. And because sound waves and magnetic fields can penetrate deep into our tissues, so you have these beautiful pictures of the human fetus or the human brain. But the problem is that historically, while these technologies have been good at giving us anatomy and maybe some physiology, they don't tell us anything about what cells are doing. Okay, so the mission of our lab, since we started here at Caltech, has been to take the physics of sound waves and magnetic fields, take the technologies on the right, and extend their abilities to the cellular level through biomolecular engineering, meaning that just like that GFP, green fluorescent protein, was found in jellyfish and adapted for fluorescent imaging, we've been looking for and engineering proteins and other genetically encodable materials that cells could make that instead of light can interact with sound waves and magnetic fields. Okay, and today I'm going to tell you a little bit about our work on ultrasound, which we love because it's both very simple and at the same time powerful. It's simple in the sense that ultrasound imaging is just based on sound waves that we transmit into a tissue that propagate through the tissue until they encounter an interface with something that has a different density or stiffness, like bone versus muscle. Okay? And at that interface, some of that sound wave we transmitted will get reflected back. And based on how long it took between when we emitted the sound wave and when the echo came back to us, we can tell how deep that interface is. Okay? And then we can just sweep the beam left and right and get an image like this one on the bottom left. And you can tell right away, ultrasound can go deep. This is a couple decimeters into the body. What I think is harder to appreciate just from these normal ultrasound images is it's actually a pretty high-tech technique. So today in our lab, without any special equipment or just using ordinary stuff, we get spatial resolution deep inside tissues on the scale of 100 microns. So that's 1/10 of a millimeter, which is not quite at the level of a single molecule or single cell. But if you think about how tissues are organized in the liver or the brain or in the gut, a lot of things are actually organized on this 100 micron to a millimeter scale. So it's already really good. And on top of that, people recently have developed even more advanced techniques like super resolution ultrasound. So in this image here in the middle, we're looking at a rat brain where all these little squiggles are blood vessels inside the brain. This is imaged in a living animal where they're resolved at 10 microns, so 1/100th of a millimeter resolution, which is remarkable for live imaging in this kind of scale. And while being pretty high performance, ultrasound is also relatively accessible, meaning today, for about $5,000, you can buy an ultrasound system where all the electronics and signal processing is contained inside this handle, and you can hook it up to your iPhone, and you're off and running doing ultrasound. So in principle, every doctor and biology lab in the world could be using ultrasound if it only did what we wanted it to do. But the limitation, again, is that so far from what I showed you here about ultrasound is imaging anatomy, right, where the bones are, where the muscles are, maybe some physiology here by looking at blood flow, but it's not telling us about what cells are doing. And so the missing piece here is there a green fluorescent protein, quote unquote, for ultrasound? And just to make the analogy a little bit more explicit, so what happened with fluorescent proteins is before that there were chemical fluorophores. These are chemical molecules that can fluoresce. And GFP was found as something that is made by a cell that has similar properties to these dyes. And for ultrasound, the synthetic imaging agent that already existed is called microbubbles. This is something used in the clinic. And as the name suggests, these are micron-sized bubbles of gas, typically stabilized by a lipid shell. And because they have a lower density and are more compressible than the surrounding blood or tissue, they are really good at reflecting sound waves. So if you inject these microbubbles into the bloodstream, you can see the vasculature very nicely. And in fact, that was used to generate this beautiful image here I showed you earlier. And so the question is there something in nature that is genetically encoded that has similar properties to microbubbles? And this is where we got really lucky, and Mother Nature gave us an enormous gift, which came from photosynthetic organisms, similar to the ones that you might see when you're landing in SFO with the South Bay salt ponds, where the color in these bodies of water is coming from photosynthetic microbes that had to solve the evolutionary problem of learning how to float because they want to be near the top of the water to get enough sunlight for photosynthesis. And the way they evolved to do that is by forming these really beautiful intracellular self-assembling protein nanostructures called gas vesicles. Okay, let me just slow down for a second. Intracellular, made inside the cell, self-assembling from protein components that the cell makes based on genes. Nanostructures, so this is an electron micrograph of a gas vesicle structure that we purified from a cyanobacterium. And you can see from the scale bar here, this is about 100 nanometers in size, so really, really, really small. And what's special about these little protein nanostructures is that they're hollow on the inside and filled with gas. Okay, so imagine a ping pong ball that's now been shrunken down by orders of magnitude, and it has a shell that's very thin, so 2 nanometer thick shell, and has gas on the inside, air, typically things that are dissolved in the surrounding media. And when these cells want to float higher, they turn on the set of genes that allows them to form more of these gas vesicles. The cell becomes less dense and flows to the top. And when they want to sink, they can dilute these out with cell division or break them down with enzymes and so on. So just a really remarkable piece of evolution. Now, some of you who are kind of more, this is what a cell looks like when it's full of these things. Some of you might be wondering, like, well, how the heck does this thing actually exclude water, and how does it contain air? So I thought I'd just give you one slide on this. And I actually didn't know. So these structures evolved more than 1 billion years ago. Their existence was discovered by German scientists in 1895. We engineers got our hands on them, like, now I'm sad to say, like 13 years ago. And we only found out their atomic structure in 2023. Okay, so this has like been a long time to do, but now we know. And what's cool about these things is that if you look at a gas vesicle, so it's this cylindrical shape with cones on the ends, you zoom in, you see it has these like ribs kind of structure. You zoom in to the atomic level, and what we see is that the part of the proteins that make them up that face the water are what's called hydrophilic, meaning that they like to interface with water, whereas the part that is facing the interior is extremely hydrophobic, meaning it hates to interact with water molecules and excludes them. And that is what prevents water from coming in and forming a liquid phase on the inside of these structures. So things that we use in our daily lives, like Gore-Tex and stuff like that are water repelling, nature figured out how to do that 1 billion years ago and used it to make this. Okay, anyway, so we hypothesized that because you have these little proteins filled with air, that we might be able to see them with ultrasound. And so this is data from the first paper we published on this back in 2014, where what we did is we purified some of these gas vesicles. We embedded them in hydrogels, which are acoustically transparent. So that's the black background you see in these ultrasound images. And then here we have a series of concentration of gas vesicles where we're imaging the ultrasound and we saw ultrasound signal. So this was exciting. Now we have a protein and we're able to see it with ultrasound. And of course, one of the first questions was, can you actually see it inside of a living, breathing animal with all the movement and noise and so on? So we took some mice and we started injecting them into their vasculature with gas vesicles. And when you inject nanostructures into the bloodstream, most of them get taken up by the liver. That's part of its job, to clear these things. And so we're going to look here at a video where this is a mouse. This is the liver of the mouse at the top. And we're going to start the video. And at some point, we're going to start injecting the gas vesicles. Okay, so watch the liver here at the top. We're starting the injection. So keep watching. And over time, we'll see the accumulation of signal in the liver. Okay, and this was exciting because to our knowledge, this was the first time that a biomolecule could be seen non-invasively with ultrasound inside of a living, breathing animal. And this opened up a new field of research that we call biomolecular ultrasound, which has been really, really fun just to see how far we can push it. And Caltech's been the perfect place for this because here we had to solve not only protein questions and synthetic biology questions of how do you string these genes together and make them work in different kind of cells, but also mechanics questions. Because the way these little nanostructures respond to ultrasound is by buckling and unbuckling under the sound wave that's pictured in the bottom left. This is stuff I had no idea about, but it turned out that our mechie and aeronautics colleagues think about all the time. And so we published some beautiful papers using their modeling methods to understand what's going on in our system. So I want to show you a few snippets of what this has looked like and where we are now. One of the biggest questions, and frankly areas of skepticism when we first put forward this idea, was whether we could ever take these genes from the cyanobacteria and move them into animal cells. Can we express gas vesicles in animal cells? And the reason for the skepticism was people said, okay, well, with green fluorescent protein, you just have one gene that's making one protein. It's very simple. People figured that out. Here, you have a group of genes that work together to assemble this more complex structure. And so this is like a set of genes up here that encode gas vesicles. And you can think of some of them as the bricks. These are the proteins that end up being part of the structure. And you can think of some of these other genes as construction cranes and construction workers. We call them chaperones and assembly factors that put these bricks into the right place to form the structure. And you need to have the right ratio of cranes and workers to bricks to have the house look right at the end. And until we started working on this, people hadn't really tried to have very precise ratios or stoichiometries of genes that they're transferring over from one domain of life to another. So we had grant proposal reviewers and journal editors that said, yeah, this is all really cute, but you'll never get this to work in this way. Luckily at Caltech, the harder the problem, the more the PhD students want to work on it. And so here I'm going to summarize 4 years of work by Arash and Gabrielle with this little arrow here, where what they did was basically take all these genes from cyanobacteria and figure out how to string them together into genetic code that animal cells can read that encodes all these genes at the right ratios. And I'm not going to go into details, but just point out what's cool about this is that in making these genetic cassettes, we're borrowing tricks from all over nature. Some of the stuff comes from human cells themselves. Some of it comes from viruses, because viruses also have this problem of how to efficiently encode things that you can put into an animal cell to make new stuff. So we borrowed a lot of things from synthetic biology, and now we have a set of genetic constructs that we put those into the genome of a human cell, like the HEC cell here. They can start to form gas vesicles. This is, again, an electron micrograph where you see these bushels of gas vesicles inside. And more importantly, now when we image these live cells under ultrasound, we see ultrasound signal. And here we connected it to a very simple genetic program where we're giving this drug and it's causing expression. And the more of the drug we give, the more ultrasound signal we see. So that was an important step. And of course, we also wanted to see this in vivo in a living animal. And initially, we did these experiments where we formed little tumors under the skin of the mouse. And we engineered those tumor cells so that they can produce these gas vesicles. And what's known about these tumors as they grow is that they tend to be better vascularized, have better blood flow towards the perimeter than they do towards the core of the tumor. And therefore, the cells towards the perimeter should be better at expressing these gas vesicles and therefore producing ultrasound contrast. And that's something that would have been very hard to see optically, because if you just look at light coming out of a tumor, it would just look like a glowing blob. But we were hoping that with ultrasound, we'd be able to see this kind of pattern that I just described. And luckily, that turned out to be true. So the image you're looking at here is in grayscale is the anatomy. So this here at the top is the skin, and there's like a little elongated tumor underneath. And in red scale is nonlinear ultrasound signal that's coming specifically from our gas vesicles. And we saw it looked like this elongated oval structure where there's more signal towards the outside than there is towards the middle. And we can confirm this by taking that very same tumor and then taking it out, sectioning it, and looking at it under fluorescence, and we see this very nice correspondence between what we saw with ultrasound and what we see with light. The difference being that the image on the right is after the mouse has been killed and cut into little pieces under ethically approved protocols. And on the left is the ultrasound image where we can continue to follow the dynamics of what's going on. So the kind of stuff we're doing with this now is like getting into more sophisticated experiments. For example, we want to learn about metastasis, which is actually very hard to study in animal models because you can't tell where did the metastasis go and where is it forming. So there's just some images where we're looking at a mouse liver. It's getting metastasis in the liver. The grayscale is the anatomy, and these little green dots are signal that's coming from tumor cells that are expressing gas vesicles. This is a 3D image, so you can see these little green dots. Now, what's cool is we can take the same mouse and look at it a week later. And now we see that some of these metastases are starting to grow. Some of them are disappearing because, as we now know, they're being taken out by the immune system. And by week 3, now the liver is starting to get uncomfortably full. And so now with ultrasound, we can study this biological process and use it to help us understand fundamentally what's going on and how these things would respond to different drug treatments. I also said that we want to follow cell therapies, and I gave CAR T therapy as an example. And again, here, these are immune cells that are taken out of a patient, modified genetically, and sent back in to fight a tumor. And what we did is we modified these cells, so human T cells, so they can also produce our acoustic reporter, so you could see where they are. And now when we take images in mice with tumors, after we inject these cells, we get signal that's coming not from the tumor cells, but coming from the immune cells that infiltrated into the tumor. So we're tracking that they got to the right place. Again, have very good correspondence between ultrasound and what we see optically later. So this means we're seeing where they are in the tumor. And our ultrasound signal corresponds nicely to the number of these T cells that are actually present in the tumor. So if you're trying to track how well is the therapy going, are these cells getting there and proliferating, this could be a way to do it earlier than just seeing did the patient die or live. And maybe you could adjust the therapy better from that. Another thing I advertised in the beginning is using cells, probiotics in the body. And so we said, let's turn this into a diagnostic. In particular, if you have gastrointestinal inflammation, if it gets severe enough, at some point you want to know where is it? What segment of the colon, for example, is it, and how bad is it? Now, how is that done today? Not very comfortably. You send in a probe, colonoscopy, and see what's going on, and that's very uncomfortable. And so we said, let's make it easier by making a diagnostic yogurt. Okay, so we're engineer probiotics, so you can ingest them. They'll go into the GI tract where they'll stay for a short amount of time, a couple days. Wherever they sense inflammation molecularly, they will produce ultrasound signal. And then instead of a colonoscopy, you could just do an ultrasound scan, so it'd be much, much better. So we engineered probiotics with a genetic program where in response to specific inflammatory biomarkers, they produce gas vesicles. And so I'm just going to show you some summary results from this. I'm going to show you 3 mice here. They're images. The mouse is like on its back, and you can see the abdomen. And hopefully you can tell from these 3 mice which one has inflammation. It's the one in the middle. And it looks even nicer in 3D. Oops. So this is rotating of the torso, and you can very nicely see the cecum and some of the colon here where the inflammation is taking place. Okay. Last thing I want to show you, this is to embarrass George, who's the student that'll be in the video that you'll see, is what can you actually do with purified gas vesicles? So what I showed you so far is we're imaging cells, but since we already have these little nanostructures, maybe there's things we can do with them in purified form as well. And there's this idea that George and Claire, who was a postdoc in the lab, had of whether if we inject them into the bloodstream, they might get accumulated in tumors because they have a leaky vasculature, and maybe we'll be able to see the tumors nicely with this. And I told them, like, nah, these structures are too big. They'll never get out of the vasculature. I'm very skeptical. And of course, they completely ignored me and did the experiment. So the video we're going to see here, this is a mouse brain in cross-section. The mouse has a tumor in its brain. We're going to start the video where the colorful stuff is going to be coming from the gas vesicles. Now, unfortunately, this video, which we submitted to a journal, has the tumor outlined before you start seeing the accumulation of signal. So if you don't mind, just close your eyes for like several seconds, and then I'll tell you when to open them. Okay, so close your eyes. Thank you. Keep them closed, 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 closed. Open. Okay, now we're injecting gas vesicles, and we're going to start to see some signal. Initially, it's widely distributed, but over time starts to get concentrated. Okay, so hopefully you can tell me from this video where the tumor is located. Okay, and in fact, it turned out to work better than we expected. And so we now have this concept of using acoustic tumor paint, meaning we inject these things systemically, and then when you want to see where the tumor is, you can do it with ultrasound. The correspondence is very nice between what we see with ultrasound in the living animal and subsequent histology. And we know even a little bit about what are the molecular mechanisms by how these things get taken up. And so George, who's an MD PhD student and aspires to be a neurosurgeon, said, let's use this to guide tumor resection. And this actually is even more difficult, apparently, in mice than it is in humans because they're so small. So they cut out these tiny tumors. But he showed that it's doable by first we can see where the tumor is under ultrasound, and then he cuts it out, and then post-resection we can see that the tumor is gone. And this corresponds very nicely to gold standard MRI imaging. Okay, I see I'm running out of time. So I'm going to skip to the video where George describes this. Sorry. And we're going to end with that. So here's George.
Hi, I'm George Dagalian, and I am a graduate student in the Mikhail Shapiro group here at Caltech. Our lab focuses on using ultrasound as a way to image and interact with things deep inside the body.
Wait, sorry. Is the audio okay, or do we need to do something?
No.
Can I control that from my computer, or is that something?
It's understandable.
It's understandable? Okay.
That close, but
Okay, all right.
My project specifically focuses on using ultrasound as a way to initially diagnose and guide surgical resection of brain tumors, and then be able to monitor them in a frequent and longitudinal manner. And this is something that deeply resonates with me, and I'm grateful to be working on because unfortunately my grandfather passed away from brain cancer. So I want to actually be able to show you some of the work that we do, so why don't you guys join us? In the Shapiro lab, we do a wide array of things. In this bay, what we're doing is we're studying the trafficking of immune cells to tumors. Here we're studying the effects of ultrasound on neurons as a way to control them deep inside the body. And here what we're doing is we're purifying gas vesicles from cyanobacteria. These gas vesicles are the bread and butter of our lab. They are gas-filled protein nanostructures that look like tiny footballs. And in this research, they are critical for high-contrast imaging. My colleague Trevor Foshman takes cultures of cyanobacteria and grows them out in large incubators in the Krellin lab. Cyanobacteria produce these vesicles to help them float to the surface where they can photosynthesize. So we can actually harness that to do buoyancy purification. We spin down the cells to separate the gas vesicles from cellular debris. When they come out of the centrifuge, the vesicles are white and float to the surface. The excess fluid is then removed, leaving the purified and concentrated gas vesicles ready to be used for our experiments. These gas vesicles are organic, biodegradable, and can be collapsed with high pressure. In our lab, we have cancer cells in these large vats that are frozen down. What we'll then do is we'll take these frozen down vials and thaw them. We'll take them inside these biosafety cabinets and grow them out on these plates. We'll incubate them in the incubator for a long period of time until we're ready to harvest them for the next step of our experiment. Now let's head over to our Caltech Brain Imaging Center.
We have to complement the
Once the brain tumors are large enough, we can image the brain using ultrasound. Here you can see a Doppler image of the blood vessels in the brain. As it can be seen, it's kind of hard to tell where the tumor is versus normal tissue. But the moment we start injecting these purified gas vesicles with a simple IV injection, the entire brain lights up, but over time, it really beautifully localizes to where the brain tumor is at. This localization helps guide surgical removal of the tumor and can also monitor its recurrence. A possible next step is not only using these gas vesicles as a diagnostic tool, but also as a therapeutic. Because we know that these gas vesicles localize to these brain tumors, the next question is, can we explode these gas vesicles and use it as a way to cue the immune system to attack the brain tumor?
All right, so that's George. Yeah, George. So yeah, conclusions. Ultrasound is pretty cool. And it is just becoming possible to connect ultrasound to cells and molecules using genetic tools. And there are many possibilities and challenges for us to continue to explore. I want to acknowledge the whole lab for the work that I showed. This is a picture from one of our most recent beach parties. And thank you very much for your attention and for everything you do for Caltech.
Thank you.
I think we have time for a question or two. Yes, Mason.
Just commenting question one, for those who weren't at the student experience committee, George is one of our student reps on that.
Oh, great.
So you'll be able to interact with them, not just at this lunch, but at future meetings as well. I had a very basic question, which reveals that I know nothing about ultrasound, but hopefully that's okay. Does the kind of technology for viewing these molecules change because of the scale of them, or does existing ultrasound technologies?
Yeah, that's a really great question. So luckily, existing ultrasound technology can do it. So the kind of equipment we use is analogous to the kind you'd get in the hospital. We just have the engineering versions of that where we can use computer code to program more precisely what the sound waves are that are emitted from it, but the hardware itself is the same. Yeah.
Just a great presentation and also great work. Given that the size of the gas vesicles is about 50 to 100 nanometers, and the size of a red blood cell is about 8 micrometers, and leukocyte is about tens of micrometers, do you think you are at a point where you can go to study intracellular mechanisms? For example, in a case of a red blood cell without a nucleus, how parasites can evolve over a period of time and using this technology. Or in the case of, they're already doing it with cancer in the white cells.
Yeah.
How is the resolution? How far deep can you go?
Yeah, that's a great question. I'm hesitant to say anything about red blood cells in your presence, so we can study intracellular phenomena, even though we don't quite have subcellular spatial resolution, by connecting the signals that we get from gas vesicles to things that are happening in the cell. For example, pH changes, or we've made reporters of calcium or kinase activity. Potentially, mechanics could be sensed if you somehow connected the gas vesicle behavior to intracellular pressure or other aspects there. And that's something that can be done through molecular engineering. In principle, you can go to very high frequencies of ultrasound and do cellular scale or subcellular imaging if you're doing it under microscope. But then the question is, how is that better than doing it with light? And we have to discuss that.
This is more a comment than a question. I served on the board of Royal Philips, which is one of the biggest producers of ultrasound equipment in the world. So afterwards, if you want to have an introduction there, I see you're very well connected.
Sure, yeah, that'd be great.
Because although I heard very clearly current equipment works, of course, they're looking at new generations of how do they make technological advances which could marry very well what you're doing coming from the cellular.
That'd be great. Yeah, thank you.
Great. Thank you very much.
Thank you.
That was great.